tell me it doesn't work, and every time.
Nitrogen-fixing bacteria that live in the roots of plants. help to bring nitrogen out of the atmosphere into the soil.
They're also decomposers.
They're also decomposers, so dead stuff gets broken down by bacteria. One more big one.
Carbon fixation.
Carbon fixation through photosynthesis, so taking carbon dioxide out of the atmosphere again and putting it into organic compounds that make it useful. Okay, then we started into protists, the leftovers. The things we don't know what to do with, so we just kind of group them together into a loose group. And then our loose groups still have characteristics that are iffy. So what is a characteristic that most of the supergroup Excavata share?
They have a bunch of flagella.
They can have flagella,
To a nuclei?
not Excavata. We'll get to the two nuclei guys today.
The feeding group.
They have a feeding groove on their side. And again, that's not true of all of them. And
They're aquatic.
most of them are aquatic. I don't know. Can you say 100% of them are aquatic? I think we say most about pretty much everything, but I don't know if I can think of an example that is not. Well, yeah, some of them that are parasites are not aquatic. So some of them also, two of the groups of Excavata have modified mitochondria, sort of three if you count the kinetoplastids having one giant mitochondria as being a modified mitochondria. So modified mitochondria, and their flagella that they have are structurally different than other groups. So we can kind of say that's a characteristic that they mostly have. Again, mostly is all we ever say about protists. Most of them do this, most of them do this, most of them in this group do this, and most of them do this, but we don't have a lot of absolutes when it comes to protists. And define mixotroph and then give an example of one. We'll talk about a lot more today. Well, maybe not, some more. What is a mixotroph?
They can be autotrophic and heterotropic.
So they're autotrophs and they can do photosynthesis, but they also can be heterotropic, so they can take in nutrients from the environment, whether it's through like diffusion or phagocytosis or something like that. So they can get their nutrients in a lot of different ways. This is what I would like to be. I want to be a mixotroph. I think it'd be awesome to be able to go outside and make my own food. But if I don't feel like it, I can eat something else. Give us an example of a mixotroph. Maybe somebody we haven't heard from yet.
A Venus flytrap. What
Yes,
do you think of it?
that is not a protist example, but I would agree that that is kind of an example. So Venus flytraps don't actually get their energy from the bugs they eat. They're only getting nutrients from it, but they do photosynthesize. So they get their energy from photosynthesis. So they're pseudo-mixotrophs, maybe I would say.
Is cyanobacteria
They are photosynthetic only bacteria. But if they were both, you could still say they were mixotrophs. There was one, that was the last thing I think that we talked about. By we, I mean me, because I couldn't take the count anymore. And I thought it had been like 7 hours, and it only been like 35 minutes. I really thought it had been more. The Euclidids or the Euglena that we'll look at today in lab, they're the little green guys with the little red eye spots, and they swim around, and they're really cute. We'll see some of those today in lab, hopefully. Hopefully they're alive and with us. Okay. So, on Monday we talked about the supergroup Excavata. Okay, we're going to move on to the next supergroup, SAR. I'm starting to see more and more laptops. Remember how we talked about no laptops? I said that tablets I was a little okay with, and laptops I don't love. Okay, so SAR. So I have an old edition of the textbook, and SAR isn't even there. Because they have this is new since between the 9th and 10th edition of the textbook. We keep changing the way we classify protists. So SAR is an informal grouping. It doesn't have a really super name because there's still even not agreement on this, but it gets the SAR from the three groups that are part of it. the Stromanophiles, the Alveolates, and the Rhizarians. And again, you can see these dotted lines here. Those lines are dotted because there's not a lot of agreement still. This is a phylogenetic tree, remember? So we're hypothesizing evolutionary relationships. This is showing it kind of as a monophyletic tree, meaning everything comes from one ancestor, but we don't really agree on that. So we just use dotted lines. So we're going to talk about all of these groups of the SAR supergroup. We'll talk about the 3 groups of the Stromanophiles, the 3 groups of the Alveolates, and the 3 groups of the Rhizarians, and I'm hoping that's what we'll get through today. I'm not 100% sure we'll get through all of this today. The plan is then on Friday to finish talking about protists, and we'll review because your test is on Monday. I know that no one missed that memo. Okay, no one ever misses a memo about tests being moved to a later date. So your test is on Monday. I'm working on it still. I'm anticipating, I don't know yet because I'm having trouble narrowing it down, but 15 to 25 multiple choice. And then short answer, I don't know yet. But that's all I have so far. There will probably be some diagrams, especially from like the Protists chapter, like life cycles, that kind of thing. You might see some diagrams, or say like the virus reproductive system, not system, but like cycle. You might see diagrams. I like using diagrams and having you think about diagrams. Hopefully on Friday I'll have a better picture of what it might look like and tell you a little bit more. Okay? Questions about the test? Questions about lab? Anything that I can answer now?
On the lab page, there wasn't a submission for the going further for this.
Okay, so yeah,
Printed.
Jordan emailed me about that. I would rather have it printed, so I don't remember what it said on there, and I meant to look before I came in here. I did put the link up there, but those of you who've had me before, I'm a paper person, and not in a Dunder Mifflin kind of way, but just a paper person. I don't like to grade online, so I don't do it very well, and so then things don't get graded. So there is a link there because I didn't clarify for this week. So this week, if you have a hard copy of your going further questions or you turn them in electronically, that's okay. But in the future, I'd rather just have them turned in a hard copy, and we'll talk about that again, and I'll remind you of that in lab. Today, just remember also for lab, we're going to be in our normal room. So we'll be in 214 instead of 210. I have not put up a sign yet. There will be another lab in 210, so if you go in there, you might be confused. As will they. Okay. Now let's talk about protists. Okay. So, again, there's a lot of weird words. There's a lot of pictures. There's a lot of life cycles, a lot of information. I recommend making flashcards or something like that to practice this. This week in lab, all we're going to be looking at is protists. So you'll be looking at these kinds of things again under the microscope. Lots of microscope work this week in lab. So we're going to start with Stromanophiles. Stromanophiles, stramen means straw, and piles means hair. It comes from pilos. For me, being able to break down a word helps me to think about if it helps me, if it's related to what we know about something, it helps me, I should say. So stramen means straw, and piles means hair. So the distinguishing characteristic kind of the Stromanophiles is that they have hairy flagella. So you can see in this picture up here, here's the flagella, and it's covered with all these little tiny hairs. They often also have, in addition to the hairy flagella, a smooth flagella that doesn't have hair on it. So the smooth one is shorter, the hairier one is longer. And that's where the Stromanopile comes from, hairy flagella, hairy straw, I guess. These are really important photosynthetic organisms are really important again for the ecosystem and the cycling of nutrients. They're the bottom of a lot of food chains. They're plankton. We're going to define plankton. Plankton means little things that can't control their own movement. So they float along with currents and things like that. They don't have a way to hold on to something. They don't have a way to control their movement. They're just free floating. And we'll use the term plankton a lot because a lot of the organisms we're talking about are part of the plankton. So we're talking about an example of the Stromanophiles being the diatoms. Diatoms are really a diverse group. They're also sometimes called microalgae because they're photosynthetic and they're really small. Again, when we use the term algae, it's kind of a term that we use in a lot of different ways. Algae means photosynthetic, and we say algae means photosynthetic protist. So we'll talk about lots of different kinds of algae. We'll talk about red algae, we'll talk about brown algae, we'll talk about golden algae. Whenever we say algae, we're meaning a photosynthetic protist. When we say plankton, we're meaning a free-floating organism. So these are photosynthetic plankton, so they're free-floating algae.
So plankton are
Not necessarily. Plankton is just a general term meaning something that is a free-floating organism. So it can include things that are not protists. That's what makes all this categorization fun. Okay, this is the biggest group. The diatoms are the biggest group of phytoplankton. So phytoplankton is another way for us to say photosynthetic plankton. So let's just use that term here too. So it's the biggest group of phytoplankton, and what's unique about these is they have their own skeleton, which is made of silicon dioxide. And you can see in the picture, they're kind of pretty. They have all kinds of different shapes. Like their skeleton is all kinds of different shapes. That says silicon dioxide, sorry. Dioxide. Sometimes it looks worse up there, I think, than it does here. I would define it, it's like a wall, outside wall. So I would define it more as an exoskeleton. Maybe skeleton isn't even the right word because it's a photosynthetic organism. It has a wall on the outside.
I can say because of the skeleton, do you have anything else, like micro-eyes or something like that?
Eyes, you said?
Yeah.
No. So they're photosynthetic, so they would be able to capture light, but they don't have any way to see, like a euglenid or euglena has an eye spot to detect light. They don't have anything like that. So their shell, if we want to call it a shell or a wall, is kind of like a shoebox lid. So it has the ability to expand and contract. So they can withstand really high pressures. And the book gave some crazy number that I don't remember. But they can withstand really high pressure because they have that flexibility in their two-part shell. So they've got a two-part skeleton, a two-part skeleton of silicon dioxide that allows them to survive in high pressures. They're really abundant, and if conditions are right, they can grow at really rapid rates and produce what's called a bloom. And the population of diatoms, if they're blooming, can actually affect atmospheric carbon dioxide levels. So when a diatom bloom happens and they're photosynthesizing, they can pull enough carbon dioxide out of the atmosphere that it's noticeable, which is significant. When they die, their skeletons, their shells, whatever we're calling them, their cell walls, will sink down to the bottom and they store carbon that way. So they can act as what we would call a carbon sink. The dead piles of diatoms can act as a carbon sink, storing carbon that they've taken out of the atmosphere at the bottom of the ocean. But their growth is limited by things that, by elements that are present. So one idea to deal with that some people are floating around, scientists, not just people, is the idea of iron seeding the ocean, because iron is the limiting reagent, it's the limiting element in the growth of diatoms. So if we give them more iron, they grow faster, and when they grow faster, they take more carbon dioxide out of the atmosphere. So one idea that some people are floating is, let's dump a bunch of iron into the ocean, cause these diatom blooms on purpose, so that they'll take up carbon dioxide and take it out of the atmosphere to reduce atmospheric carbon dioxide levels. Some people think that's a great idea, and some people think maybe not such a great idea, because we don't really know what are the consequences of us dumping large amounts of iron into the ocean, but long term or a large scale. But on a small scale level, it does work.
So you think it's good?
I didn't say that. I said on a small scale level, it works.
So large scale, what's your opinion on it?
I think that anytime we start talking about messing around with big systems in a large way, I think it's a concern, because we don't know what are the other effects. So we dump a lot of iron into the ocean. What else is that going to do? How long is it going to last? Is it worth it? I don't know. I'm not saying it's bad. I'm saying I don't know that we know enough either to make that kind of decision. So as they die and their shells go to the bottom, they're also eaten by a lot of organisms, because again, they're the bottom of the food chain, but the shells are not digested, so those end up at the bottom of the ocean. There can be layers of diatom skeletons at the bottom of the ocean that are hundreds of meters deep, and we actually mine that to get what's called diatomaceous earth, which you'll look at today in lab. Diatomaceous earth is used as a filtration agent. So, like, we have a pool, and we use DE, diatomaceous earth, the little skeletons of these guys, to filter our pool. It's just crazy. You may also, at some point, have, if you ever look at your toothpaste ingredients, maybe you don't, I don't know. Sometimes when I'm brushing my teeth, I read my toothpaste ingredients. And one of the things that you'll see is hydrated silica. It's the shells of the diatoms that you're brushing your teeth with to scrub them clean.
I know another use for diatomaceous earth is often like if you've got trash with your plants, you can sprinkle it on the floor.
Yes. So that also, they can be sharp. So if you want to kill like snails or something, or slugs, you can just sprinkle this on the ground and they crawl over it and it cuts them up and kills them. I think it's sad. I agree. Okay. Those are diatoms. So today we'll look at diatomaceous earth in lab, and I think we have some prepared slides. It never looks as pretty as it does in the picture. I'm sorry. These are like the selection of super pretty diatoms. Ours is just like regular old diatoms. I don't know. Or they're crushed or something. Okay. So the next one, if you took the slides for Moodle, I split it into two because I decided we would need more room to write. So the next group of Stromanophiles that we're going to talk about are golden algae and brown algae. So golden algae is on this slide, brown algae is on the next one. Golden algae is named that because what does algae mean? It's photosynthetic. It's a photosynthetic protist. And it has kind of a golden color. So it has two kinds of photosynthetic pigments called carotenoids. Now I'm doubting my spelling. Okay.
How you see that word?
Carotenoids. So they have yellow and brown carotenoids. All of them are photosynthetic, which I'm going to abbreviate, but we can't ever say all because we're talking about protists. Some in this group are also mixotrophs. So they're still photosynthetic, but in addition to being photosynthetic, they're also heterotrophic. The golden algae are often, again, we don't use always. They're often biflagellated. So they usually have 2 flagella on one end. And again, most are unicellular, but the one you see in the picture is not. That's a colonial golden algae, so they can live together in groups and kind of start to cooperate. So again, remembering when we're talking about unicellular organisms, they have to do all the functions of life in that one cell. We're multicellular, so we have specialization, so some parts of our body do some things and some parts of the body do others. So some protists have found ways to join together to be colonial so that they can start to sort of specialize and share the work. So that's a colonial golden algae called Dinobrium. Dinobrium.
When you say like a colonial algae, is that similar to like a biotome? Is that true?
I think you could say that, yes. So I mean, it's just a group of them together, and again, kind of sharing the workload. Sometimes some specialization, sometimes not any specialization, just working together. And again, these guys are part of plankton. So plankton being stuff that's just floating around. Can't control where it's going. Usually plankton lives near the surface. Why would it make sense that algae and diatoms would need to live near the surface? If they're photosynthetic, they need to have sunlight, and sunlight can only penetrate the water so far. So most plankton is going to live at the surface level. I don't think we have any golden algae to look at, sorry. We might have one golden algae to look at. The second part of Stromanophiles, in terms of colored algae, are the brown algae. Brown algae have brown carotenoids. That's their that's their photosynthetic pigment, are the brown carotenoids. These are different, and we're going to use a word here that we don't usually use. All of them are multicellular. I know. All of them are multicellular. These are often what we refer to as seaweed, if you want to talk about another generic term that we don't really understand, or that we use in lots of different ways. These are marine seaweeds, so they're saltwater seaweeds. And a lot of them have, because they're multicellular, they have specialized structures. So you can see in the picture, this is a brown algae. A lot of them grow close to the shore. So again, close to the surface, but close to the shore where there's crashing waves. So they have to be able to withstand being pushed around all the time. And they also have to be able to withstand being covered with water, but maybe sometimes not covered with water because of the change in tides or the way the waves are crashing. So they have specialization. This part of the brown algae is called the holdfast. Guess what it does? It holds it to the then the stalk part is called the stipe. And then these leaf-like things are called blades. They have a cell wall that is made of cellulose, like a what? What other organisms have cellulose cell walls? It's a whole kingdom that has it. Plants. So they're similar to plants in that they have cellulose, but they also, in addition to that, have a polysaccharide gel. that helps to prevent them from drying out. Some of these are eaten. So some, like, this is not it. I think this is a kelp, but laminaria, which is one we'll look at in lab, is eaten. People eat it plain. Some of them are used to produce what's called algin, which is a thickening agent. So that's used in things like salad dressings, puddings. It's a natural thickener that comes from algae, from brown algae. And they have a life cycle that is referred to as alternation of generations. So they have both, here's a little genetic stuff coming back to you. Haploid, which we use the N for, and diploid, multicellular generations. So there's both haploid and diploid multicellular generations. So for humans, we're diploid. We make haploid gametes. The haploid gametes fuse to make a diploid zygote. So there's no multicellular haploid human things. Okay, these guys have that. And they have what's called a sporophyte and a gametophyte. So the sporophyte makes spores, which are haploid. And the gametophyte makes gametes, which are also haploid but then fuse to become diploid. So your book talks about this some. Another term that we use often when we talk about alternation of generations is heterotropic. Nope, sorry, heteromorphic, sorry. We also use the term heterotropic, but not related to that. Heteromorphic meaning different body forms. So the sporophyte and the gametophyte look different from each other. Sometimes the sporophyte and gametophyte look similar enough that we call them isomorphic, but these guys are heteromorphic. So the diploid and haploid generations look different from one another. Okay. Oh my goodness. Okay. So that was one group of the SAR group. That was the Stromanophiles. The second group, again, we're breaking down into smaller groups, are the Alveolates. And we're starting with the Dino or Dinoflagellates. I had a discussion with Dr. Rousseau about this yesterday. He says Dinoflagellates. I think I say Dinoflagellates, but when I search that myself. So either way, I don't know what you mean. Dino or Dinoflagellates. So these are alveolates. So when we talk about alveolates, we're going to use the word most again because we don't want to make any definitive statements. Most of them have alveoli. Oh my gosh. Too close to the edge or something here. It's making my handwriting even worse. So most of them have alveoli, which you might be familiar with. You might have heard that word before. Not ravioli. It's not lunchtime yet. Alveoli, what part of your body has alveoli? Your lungs. They're the little sacs at the end of your trachea splitting into bronchi, splitting into bronchioles, where gas exchange actually happens. So it's the same idea. It's like a little sac. a membrane-bound sac that's under the plasma membrane, and we don't really know what it does. Isn't that great? I think that's great. So some people think that its function is to help like regulate water levels, like tonicity within the cell. Some people think it has to do with ion content, kind of like kidney kind of stuff. Some people think it has to do with stability because they're found around the flagella. So you can see here it's at the end around where the flagella is, so it might help with stability. We don't really know. Most of them have it though, most of them. So dinoflagellates, I think I have one more picture. Oh, not that many pictures. Dinoflagellates, not surprisingly, have flagella. They actually have 2 flagella. I think we're going to say all. All dinoflagellates have 2. And you can see here, it's kind of got this groove through the body. So one of the flagella wraps around that groove, and then the other one sticks out. So one wraps around it, and one sticks out. So you've got this one sticking out, and then the one wrapping around. Because of the way their flagella are arranged, when they move, they spin. So they can't like swim straight, they just spin around. I'll show you a video in a minute. If you ever want something soothing, you can watch dinoflagellates spin around. There are many videos on YouTube of them with background music. The other thing about them that is a common characteristic of the dinoflagellates is they have cellulose plates as part of their cell wall. It's like an armor for them. Their cell wall is made up of cellulose plates. And they're present in plankton, like everything else we've talked about so far today. They're present in plankton, so they're free floating, although they have a little bit of ability to control their direction. Some of them are phytoplankton, so we have some that are photosynthetic. So here come the sums again. Some of them are mixotrophs, and some are heterotrophs. Most of them, the photosynthetic ones and the mixotrophs, would be considered to be algae. They also have, so remember when we were talking about DNA structure, we said the DNA could be loose or condensed. Dinoflagellates always have condensed chromatin, so their DNA is always tightly wound up. And lots of them function as what we call endosymbionts. So they can actually have other little organisms that live within them, and everybody's happy. Symbiosis. They're working together. Everybody's happy. Endosymbionts. Yeah, endosymbionts. Endosymbionts. It means that they can live with other organisms, like other organisms can actually live inside them, or they can live inside other organisms. So we talk about a lot of general characteristics. We give you a couple examples, but there's actually cool things about some of these organisms too that are not necessarily, I shouldn't say, they make it a little more interesting. Some of these are bioluminescent. So they have the ability to actually make light. Now I don't remember which one is.
You said only some, right?
Yep, only some. Are you not going to do this one? Did I not? I know, I made a whole bunch of dots around it, and I'm wondering if it's because I'm in this weird mode, this weird way to view it. I can't click on my, but maybe I didn't make the link. Is this one going to work? That one's going to work. I didn't make a good link. Now I will never be able to find the right video if I wait right now.
Booked a sunny Virgo ski chalet with endless views of snow-covered peaks. But the thing about
Ha. So this is showing you they only produce light like when they're moved, when they're disturbed. So this is showing you a surfer. And yeah, when the waves crash, that's Dinoflagellates. How cool is that?
Are there other parts of water that are in California that have this?
You can see it in lots of different places around the world.
Like where?
You would just, more warmer water, although California water is not always warm. This is California. This is San Diego, which is a warmer part.
Are there any on our coast?
I think so, but probably further south. Yeah, I mean, I've heard of it in the Caribbean, but if you have to kind of be there at the right time. It's not like it doesn't always happen. There have to be, the population of Dinoflagellates has to be high enough. This is like amazing, and this is a, like, this would be not what you would normally see.
When this does happen, you can actually develop in person.
Have you seen it?
Like, no, I'm saying
Oh,
when that
yeah.
happens, it's something you can actually see rather than like.
Yes.
Okay.
Yes. You can actually see it. So this, like, again, this is phenomenal. But you can Google bioluminescent and you will see it. Like, it's just there.
What causes them to like light up?
The way that they move, they can release when electrons move, they can release, it releases light, kind of like a glow stick kind of reaction. Like when you crack the glow stick, that causes the movement of electrons from one shell to another shell, and that releases light, and it's the same kind of thing for them. The other thing that they're famous for that isn't as cool as being bioluminescent, we talked about diatoms having blooms. They can cause we can call red tides. What are called red tides, which are again a bloom. It's a period of really explosive growth of dinoflagellates. And the pigments that they have in them, the carotenoids that they have in them, are kind of reddish or pinkish. So they can make the water, as you can see here, look red or pink. And that's not actually good, because they also produce toxins. And so the toxins that they produce are taken up by fish. Then people can eat the fish. You can get sick. You can die. So bad things about dinoflagellates. But it's especially a problem for fish and shellfish. Shellfish are filter feeders, so they're filtering out things like dinoflagellates. So if they're in a red tide area, they're going to filter out the dinoflagellates. We hear a lot about algal blooms in lots of places, or maybe you don't. Maybe I just do because I'm nerdy. But Lake Erie had a really big problem with algae blooms probably 5 or 6 years ago. That's not dinoflagellates. Okay, again, algae is kind of a generic term. That actually is more of a cyanobacteria problem in Lake Erie. Red tides are a marine problem, so it's a saltwater issue. But the algal blooms in like Lake Erie are caused by cyanobacteria, and that's also a problem. This happens often because there's an influx of nutrients. For some reason, there's a lot of nutrients in the water. It can be from fertilizer runoff. It can be from all of a sudden the sewage treatment plant isn't working. You know, lots of different things, but can cause an influx of nutrients. But influx of nutrients causes excessive growth. And with dinoflagellates, we worry about red tides. go to the next slide. What? There we go. I got it. I counted. I think I only have 8 more classes. I figure by the end of this, I'm really going to be a professional at this, and it won't matter anymore because I won't need it anymore.
She's coming back in 8 more classes.
I think so. Because she's coming back after February break, and we only have 8 more classes before February break, and that is totally crazy. Okay. So another alveolates group. So we divided the alveolates group up. The other next alveolates group are the apicomplexans. And we're gonna, I don't think there's any way to talk about these in a good light. Okay, they're all just bad.
We're all bad.
Apicomplexans, I shouldn't say all, most of them are parasites. I made a blanket statement and I shouldn't have. Most of them are parasites. Oh my gosh, this stylus is just done with this whole thing. That says parasites. I think on your test I'm just going to write words and see if you can read them. And that's going to be your whole test. That says parasite. I think my stylus has a parasite. Most of them are parasites. And they have a pretty complex life cycle, as you can see here, that involves both asexual and sexual reproduction. So asexual and sexual reproduction that require 2 hosts, not just 1. So asexual and sexual reproduction and 2 hosts. So we're going to talk about this one. This example is Plasmodium. Anyone know what Plasmodium causes? It does. It does burst red blood cells, as it causes malaria. So this is Plasmodium. Which is the causative agent of malaria. Malaria infects 2 organisms. What are the 2 hosts for malaria? Mosquitoes and people. So you have to have both of those for them to complete their life cycle. So we're going to go through this starting inside a mosquito. And I don't have to write much here. That's a good thing. So it's a cycle, so we're just picking a place to start. So we're starting with this Sporozoites. N meaning what? What does N mean? Haploid, meaning half the number of chromosomes of a normal cell. So, a mosquito bites a person. You can see there's actually a little mosquito here biting someone. And when they bite you, they inject some of their saliva into you. And so the sporozoites are injected with that saliva. So they pass into the body through the saliva of the mosquito. And they head for the liver. So through the bloodstream, they're going to head to the liver. Once they're in the liver, they're going to go into the liver cells and reproduce. So they reproduce in liver cells, so that's a specific, it's not just people, it's liver cells of people. So they reproduce in the liver cells, and then they are released as merozoites, which are still haploid. So the merozoites then enter red blood cells, where they're undergoing asexual reproduction, and it takes 48 to 72 hours in here for them to reproduce enough that they can burst out. During this time, this is when people start to feel the fever and chills that are characteristic of malaria while they're reproducing in the red blood cells. They can burst out and go infect more red blood cells. So some of them will go back and infect more red blood cells. Some of them, now they're called gametocytes, some of them are going to make their way through the bloodstream to, what's this? A mosquito. So the mosquito, when it bites you, not only injects saliva, but also sucks your blood out. So it can, when these gametocytes are released into the blood, they can be sucked up by a mosquito. Okay, when they're sucked up, then they form male and female gametes, which then fertilize. This is all happening inside the mosquito. That forms a zygote after fertilization. Then meiosis occurs, producing an oocyst, which then bursts, releasing more sporozoites into the saliva of the mosquito. This is happening in the digestive tract of the mosquito, and it can keep going around and around and around and around. But it requires both hosts to have this happen. And we'll have to finish talking about malaria on Friday. So Friday we'll finish talking about the SAR, and we'll talk about archaeplastids, and hopefully have time to review. Some of you I will see at 1 o'clock today. Some of you I will see at 1:40 tomorrow. Hopefully. Unless Scott Hesko is right. Depends on whose forecast is right.